Hello, this is Dr. Marino Estrella, and I'm making a video about molecular diffusion and solutes properties to diffuse. This video is dedicated to my med students at OEM of the course of Basic Concepts of Medicine. Okay, so this is a video elaboration that came out of a survey that I sent it to my students, and this was the most frequent response that they wanted a video elaboration for molecular diffusion and solute properties to diffuse. So this is for you guys. Okay, so molecular diffusion can be subdivided in what is micromolecular diffusion and macromolecular diffusion. The micromolecular diffusion can be subdivided into passive, non-mediated, or simple, and a good example of this is the oxygen and the CO2. And we also have the passive mediated or facilitated diffusion, and that one can be subdivided into the transporter mediated. And one good solute that uses this type of mediation is the glucose. And we also have the channel mediated, which a good example of solutes that use this type of diffusion is the ions. Okay, so remember that ions can be positively charged or negatively charged. The positively charged are the cations, and the negatively charged are the anions. Okay, so the active diffusion is the type of diffusion that moves against concentration. So remember, both passive non-mediated and passive mediated, they are both in favor of the concentration. Now active diffusion is the only one that is against the concentration. So the active diffusion must use pumps, and this pump uses energy in order to diffuse the solute against the concentration. Okay, so pumps can be also classified in B-type pumps, and in the B-type pumps we have the hydrogen pumps. We got the P-type pumps, and in the P-type pumps we have the sodium potassium pumps, and in the ATP pumps, ATP binding cassette pumps, we have the multi-drug resistant protein family. Okay, so those are the pumps that we have, but here I have an example, the sodium potassium ATP pump. Okay, for micromolecules, this video is actually more orientated to micromolecules, but micromolecules can be endocytosis and exocytosis basically. Okay, so the difference between channels and transporters is that channels are faster. Okay, channels are faster than transporters, meaning that they can intake more solutes than in a more rapid manner than transporters. Okay, but channels can only move in favor of the concentration. So, but meanwhile, transporters can move against concentration using the pumps. Okay, so transporters are actually also pumps that just uses energy in order to move the solute against concentration. So the properties that we can talk about when we refer to the more ease to diffuse against the membrane, or the more difficult it is for that solute to pass through the membrane, we have some properties that we can talk about, and some of them are that we have unpolar, meaning that they are not charged, and we got polar, meaning that they are charged. So the more the solutes that are charged have more difficulty passing through the membrane, but the ones that are uncharged, they will pass with more ease. So ions are very difficult to pass through the membrane, extremely difficult. Hydrophobic means that they like lipids. Hydrophobic meaning that they like lipids, they don't like water at all. Okay, so hydrophobic can actually pass easily through the membrane, but hydrophilic has high difficulty to pass through the membrane. And we got the small molecules that they can pass easily through the membrane, and we got the big molecules that they have more difficulty to pass through the membrane. Okay, so let's talk about the diffusion that I already discussed, and let's explain then a little bit. So if the molecule in question, you see that on the upper side we have molecules that are in higher concentration than the lower side molecules. So as you can see, the concentration is highest on the extracellular side, which will be the upper side, and the concentration is lowest on the intracellular side. So if you move one of the circles, one of these molecules, from the upper side to the lower side, we will be moving in favor of the concentration, because we are moving from a place where you have higher concentration to a place where there's lower concentration. So you don't need energy for that. And if in case that molecule turns out to be an oxygen or CO2 that doesn't need channels or transporters, well, in that case we will call that a passive non-mediated or simple diffusion, just like I said before. Now, if the diffusion that we are talking about does need a protein in order to help them to pass from that space to the other, let's say that this is green structure here is a channel. So if you pass from the extracellular space, which is the upper side on the screen, to the lower side, which is the intracellular space in this case, you're going in favor of concentration, going where there's a lot of concentration to where there's less concentration. And since it's a channel, it will be called a facilitated passive channel mediated. Now, if you are transporting from this side to the other, but instead of using a channel, you are using a transporter now. Remember, transporters are slower. They're slower because they got to change the dimensions of the protein in order to take that solute into the intracellular side. They have to like make it spin. They gotta make it spin on the protein so they can release the solute into the intracellular fluid. So if this happened, it will be now facilitated passive transporter mediated diffusion. Now, what would happen if the movement is against the concentration? Well, you know the answer for that. I already told you what will happen. And remember that this is also a transporter, is that it will need energy. It will need ATP. So if you pass from this place where the concentration is lower, then in that case you will need ATP. And this is what happens. You got active transportation. Okay, so thanks a lot for watching, and I hope this video helps you out to study for your exam and to study for basic concepts of medicine. So have a good day, guys.